Hello again, everyone. You're joining us for another episode of Executive Platform's Blueprint podcast series. My name is Jeff Mix. I'm head of content and research. My guest today is Teresa Gorecki of Compliance Architects. We're going to be having a conversation about what is new with the FDA as the pandemic wanes, what is happening in the world of cell and gene therapy, some of the things the industry needs to understand about what's coming next. I think this is going to be a fantastic conversation. Teresa, thank you so much for joining me today.
Thank you.
I guess I want to start off with what is the FDA doing as the pandemic is waning? I think there have been a lot of changes, and the industry is always interested in what the regulating body is doing. Can we talk about that?
We can. It's interesting. So let's back up to before the pandemic started to wane. What we saw the FDA do, of course, was to stop having inspections, and they started doing a lot of 4003 requests, which are requests under new legislation to request a lot of documentation from a company. It doesn't look like they've done much with any of that documentation. They took it all in. They haven't done anything with it. So as they come back out, instead of being informed from reading all of the information that they requested, they're going back to the last inspection they did, could have been 2018, early 2019, before the pandemic hit. And so they're going back over more than 2 years of information. In many cases, they're going back over 3 or 4 years of information, which makes an inspection longer, more complex, and more difficult. So what I see in my practice is the number of documents requested is higher, the number of investigators showing up for an inspection is higher, and certainly I sense they're much more enforcement biased at this particular point in time. It's as if a few of the things that we saw emerge during the pandemic with a couple of companies that were producing vaccines have sort of fed the notion that I think FDA perhaps had some feedback that maybe they'd grown lax during the COVID pandemic to allow treatments, to come online, et cetera. So they're coming back with a very serious tone and tenor.
Are there actual changes in the regulations that the industry should be aware of that I guess maybe is meant to keep them on their toes?
I think that we've seen some changes in regulations emerge, but honestly, I haven't seen dramatic changes emerge in the last 3 or 4 years, other than all of the stuff that has emerged that allowed the vaccines and the treatments to progress quickly. So the parallel pathing, being able to submit BLAs, in a chapter-like format as you finish data and information. The 4003 request is new, where they can request a lot of information and do with it what they will. So they can write you a 4003 request and say, send me the last 2 years of CAPAs on a stick.
One of the things that I really enjoy when I speak to someone from Compliance Architects is that your organization really does help companies across the spectrum in this space. There's small startups, there's sort of the established biotechs, there's the 100-year-old pharmaceutical giants. And I wonder if we could talk a little bit about what is the difference that is happening to these different sized organizations as we come out of the pandemic and as things really sort of ramp up for the next generation of medicine.
Well, what we've seen emerge over the last 5 to 10 years in general is, FDA's willingness to grant rapid approval and rapid review, right, of novel drugs, life-saving drugs, rare disease drugs. So, in the old days, we had orphan drug status, and that got you some additional expedited review and collaboration. But with the rare disease and all of the other treatments emerging, things like CAR-T, stem cell, some of the new vaccines, some of the new treatments that are coming out for oncology, autoimmune diseases, et cetera, we're seeing a lot more willingness on the part of the FDA to be able to review things quickly. This puts a lot of pressure on the organization. So if you're a small company, what I see is that the clinical side of FDA, the reviewers, are willing to allow you to really move ahead quickly, particularly a rare disease state in children. They will do anything to help you move along rapidly. What happens when you get over to the CM&C side of the house, right, chemistry, manufacturing, and controls, is the wall goes up. And a lot of the things that you're expected to do for traditional pharmaceutical, you're still expected to do with some of these novel forms and these novel technologies. And not everybody's figured out how to adapt the regulations and the requirements to those things. And it's made it really challenging. So for the small startup company, they're virtual. They rely on their CDMOs to understand how to do that stuff. So if they don't choose the right CDMO, they're in trouble when they get to the end and they're submitting their full NDA or BLA application. In the case of the big monolithic companies, the big guys, What you see is it allows them to move ahead quite rapidly, but in a very large company, you have very set processes in development. You know, there's a time when you start validation, there's a time before that when you begin scale up. There are these prescriptive times that are all planned into project plans. There's lots of people involved. And so their ability to actually move faster when the FDA allows them is challenging for them, because they've got a large machine that they have to figure out how to retool and re-engineer to move quickly. And I would say the medium-sized companies are in a unique position because they don't have quite as large a monolithic system to drive a new product forward, but they have more infrastructure than the small virtual startup does, and they're not completely reliant on a CDMO. They have a lot of their own internal technical expertise to figure things out, and they're a little more nimble and fast and flexible.
I'd love to expand on the CMC piece you were talking about, especially as it relates to sort of the startup and very small companies. There's a lot of capital going into early stage companies right now because there's a lot of exciting things happening. But of course, there is the concern about CMC maturity. And I wonder if you could speak to that for us.
So it's very interesting because what you see in some of the newer startup companies is this lesson that's been learned through some of the companies that started up, say, 5 years ago. 5 years ago, you would have had a chief medical officer or head of clinical development that would have had an MD, you would have had a CEO, and you might have thought about supply chain. You probably might not have thought much about quality at all yet, and you had a big thought about clinical affairs and clinical development, right, and getting into the clinic. You didn't see like a VP of CMC. And the startup companies that I'm working with now that are more in preclinical or early stage development, they often early on bring in a VP of CMC, because they know that they need to start to work on how they're going to develop their technology, where they're going to produce, what that looks like. They know they have to start to work at my cell bank. Where am I going to bank it? How am I going to get a working cell bank off my master? They start working on all of these things much earlier now and thinking very seriously about it because they've watched the predecessors before them crash and burn and learn some hard lessons.
One of the things that I keep hearing over and over again is supply chain risk, supply chain disruption. I mean, it seems to be something every company has had to deal with. I can only imagine how much truer that is in the cell and gene space because it wasn't established ahead of time. I'd love your insights on that.
There's a lot of risk in the supply chain. I mean, part of that comes from COVID, right? And all the sourcing outside of the United States and trying to deal with just factories that have been shut down. But also, when you get involved in some of the new technologies from a CMC perspective, if you're doing cell culture of any kind, you're often dealing with very novel reagents in the media. Many of those are research grade. The suppliers don't want to certify them. They don't want to characterize them. They don't want to understand lot-to-lot variability. And I promise you, when you submit your CMC section, FDA is going to demand to understand that, which means the small startup has to have enough money that they can invest and buy enough different lots, enough of the all the various important research grade constituents so that they can evaluate that themselves. They need to develop additional methods to determine the stability of some of those ingredients. They need to do additional characterization studies. All of that costs a lot of money. They even have to put up stability studies on those reagents, because the suppliers that make research grade materials don't have to. Often they are small technology companies or the technology division of a larger entity, and they don't have the funding to do that either. So they're in a very difficult and unique situation, and it can cause supply chain disruption because some of the ingredients, again, are still coming from rare and interesting places, and it's hard to get them. And or it's hard to get more than one lot at a time.
Well, I think this speaks to my next question already, but I think it's so interesting that we've actually seen cell and gene therapies as one example of the next generation of medicine that are now approved, that are coming online. It's finally happening, and it's exciting. And from these early successes, there are lessons to be learned that there's a whole other tranche coming behind them that are just sort of trying to gain access and insight from the leaders and the pioneers so that they don't have to reinvent the wheel. And I wonder if you could share some of your insights there.
Oh, gosh. One of the big things I think we learned as somebody who works in quality is in the old days when you released a lot of something, it was 40,000 vials, 80,000 vials, 10 million tablets. In cell and gene therapy, a lot can be one. You get a set of cells from a patient. You treat those cells and genetically engineer them and program them to do something, and then you send them back so they go to the patient. you're faced with all kinds of challenges, like your production suite is producing one lot for one patient. The cost of that is extremely high, very, very high. So if you look at the initial CAR T therapies that have come onto the market, the treatment can be $600,000, $800,000, $1 million. Very, very expensive. So there's a big push to try to be able to do some of these novel therapies at a much lower price point and to figure out how to be able to do that. So I would say that in manufacturing and supply chain, that's going to be the leading edge for the cell and gene therapies that have come forward, is now how to produce them in a fast, flexible, easy way and make it less expensive. But the things coming forward are things like exosomes. The things coming forward are in regenerative medicine have a lot to do with how we program tissue, how we treat tissue, how we treat cells, using exosomes. I think that's going to be a very vast space. I also think we're on the fringe of some additional breakthroughs in oncology. I met with somebody that could be a potential client here at the conference yesterday who is working on precision vaccines and immunotherapy. I remove your cells. I program your T cells for your specific tumor. I give them back to you, and it's for a metastasized cancer indication, which means I'm never going to put you in remission, but the tumors will be small enough that you can live with them comfortably and live in a state of stasis. That is absolutely a miraculous thing. But the ability, the technology and the ability to do that, the level of sequencing, the kind of bioanalytical technology that we need to demonstrate that's reproducible. And how do you validate something when a lot is one? You know, when you make a lot of 100,000 or 80,000 or 40,000 and you make 3 lots over time, you've got a very large statistical basis for your data to say, okay, I can do this reliably and reproducibly. How do you do that when it's one? You do it 3 times and you're done? You know, some of the clients that I have in cell and gene or some of the tissue therapy clients I've worked with, FDA has said to them, no, 3 is not enough, because 3 was 1, a sample of 1. So it becomes very difficult because you then need to think about how do you characterize and validate your process over time while you're in clinical development, which is not how the pharmaceutical and biopharmaceutical industries think. Mm-hmm. It's very challenging, and it means you have to find the point where your process is well enough developed and it's not going to change dramatically. And at that point, you can take many more samples from these lots of 1 and do a lot more measurement and monitoring those production runs to understand that your process is reliable and reproducible over time. It's a big challenge.
I mean, not to people listen to this thing to hear from the expert, not from me, but I have been sitting in the back of conference rooms for a decade, and many years ago there was a pharmaceutical executive who says, I know this works, but I can only right now make it work for 100 millionaires and billionaires, and I've got 5 or 6 years to figure out how to commercialize it so that I can sell it into a socialized medicine system for everybody. And that is the challenge that's facing me, and I think that's exactly what you're talking about.
That is exactly what I'm talking about. And the challenge is, even though there are probably some insurance plans that will pay for things like CAR T therapy when you're a cancer patient, or maybe this vaccine that I'm referencing that's in phase 2, moving into phase 3 for certain forms of cancer, these are extremely expensive therapies, and it's very hard for me to believe that private insurance will probably pay the majority of that time and time and time again. Mm-hmm. It's harder for me to believe that socialized medicine could ever afford it. And certainly in the more highly developed nations, they're going to be able to afford more of it than those that are not as highly developed, right? Because the financial base isn't there to support the healthcare system.
Now, I know we're at the very beginning of this challenge, but of course companies are coming to you for thoughts and insights. How should they be trying to bring the cost per unit down?
This is a challenge because you've got to find ways. I mean, it's been very interesting to listen to some of the AI presentations here, although I'm not sure that's really going to be the solution. I think robotics and automation and finding a way to work with the developers so you can use very standardized equipment, very standardized, fermenters and, cell culture mechanisms. I think you're going to have to find a way to do this in a way that you can go into a suite that's a lab, and I could produce my cells in here, and you could produce your cells in there, and he could produce his cells in there, and we've developed our process so it can run in a very standardized way. I think that's certainly one way to do it. I also wonder at some point in the future if people could produce this themselves in their home.
And again, we are speaking obviously speculatively, but I had another person tell me, you know, maybe one day the answer is every hospital has a machine the size of a Coke dispenser, and the sample goes in the top and it comes out the bottom.
Every hospital does, or I see something like an alternative infusion clinic.
Right.
You know, where it could be in your neighborhood. And I also see that there are some things that could you do them in your home? I don't know.
And what a
Could you have
challenge.
something the size of a bread box on your kitchen counter and you put your cells in there and maybe somebody teaches you how to move your cells. Maybe there's a very automated way for you to do the draw. I don't know. But I think that we're not that far away from being able to conceive of things like that, because those are the things that make this much more reproducible. And then the challenges for FDA.
That's where I was going next.
Holy cow.
That's where I was going
when
next.
When you put the Coke machine, right, in the hospital.
How do you regulate it? How do you maintain quality?
And then there'll be a lot of issues, because you'll have hospitals that are really good at cleaning and sterilizing it, and you'll have hospitals that are not.
Right.
Right.
And if the bread box is in your counter, how often is someone coming around and make sure you clean your kitchen?
Exactly.
I want to pivot just a little bit to talk about Compliance Architect as a partner as companies face these challenges. I mean, as we discussed earlier, you really do work across the spectrum of size and scale and what they're doing. So if I'm a pharmaceutical executive who has been listening to this and enjoying this and saying, you know, she really knows what she's talking about, and I have a problem with X, or this is my pain point, what does getting started with Compliance Architect look like, and how would you partner with them to solve some of those challenges?
Well, it involves calling us. Certainly you can go to our website. Certainly you can call us on the phone. We have an 888 or an 800 number, something like that, right? But certainly it involves calling us. It then involves usually talking to one of our most senior level subject matter experts, Jack and myself, some of the other senior VP level subject matter experts that we have available. And I think one of the things that differentiates us from a lot of the other consultancies is they pigeonhole you into you're a project where you're going to ask for an audit, or you're going to ask for remediation, or you're going to ask for this service or that service, and you have people that are geared to that, and you have a lot of junior level consultants. We work a little bit differently. When I'm involved, I tend to talk to the client about what they need, and then I almost always understand in this market today, unless it's a tablet manufacturer or a traditional aseptic manufacturer or classic device manufacturer, I know they have a very difficult problem, and it's going to require an innovative way to apply the requirements to that problem. So right now I have a client, for example, that is a very large pharmaceutical company, but they're very heavily involved and invested in oncology. And they really are going to need to have a diagnostic product that will be part of the diagnosis of the type of cancer you have, the type of marker on the tumor that you have, to determine whether their therapy or therapies are appropriate for you or not. So as they develop their drug, in parallel, they will be developing this assay. An assay is a device. So it's not a combination product per se, but it's a very unique and challenging and complicated situation. Because as you begin stage 1 clinical development, you're going to be running assays to look at pharmacokinetics. You're going to be running various types of assays to determine whether the person should receive the drug therapy or not as an entrance criteria. So there are many companies, these are some of the large companies, that are starting to have what I call companion diagnostics. And they have to have a process that allows them to continue to develop their drug, which is well-baked, well understood, and well vetted, and well exercised, and now they've got to figure out how to run a parallel path with an assay that will eventually grow up and become a companion diagnostic, with labeling for the diagnostic that will say this goes with this drug, and the drug information will say you got to run this test to determine if this drug is right, or what the dosing needs to be, or at what point you might dose, etc. This is complicated. This is developing two things at the same time that don't get developed the same way. And more challenging yet with the new regulations in Europe, IVDR and some of the other things that are starting to come out, you need to be able to run that assay in a way that, and develop it in a way that it's been developed under design controls. Very difficult to apply design controls to the early development of an assay. The way the design control regulations were written, the way they've been commonly interpreted, and most of the experience in the industry is with what I call widgets and wadgets. You know, things that are metal, Teflon, and, you know, they're parts. They have electronic pieces to them. This is very different. And it's not like developing a traditional diagnostic assay like a test for cholesterol or a test for, protein in your urine. This is a very different kind of assay because it involves a lot of research-based agents. You're buying a lot of antibodies and a lot of other very sophisticated reagents that in their own way could almost be drugs. But you're buying research-grade materials to develop an assay with that you're going to use in part of your drug clinical development program, and your drug clinical development program and results are completely dependent on this assay. This is a major challenge in this industry for companies that they're engaging in. So, when I get involved in a project like that, I spend a lot of time listening to make sure that I understand what it is they're trying to accomplish. Then I think through the regulations, and then I enter a dialogue about what have you tried? What are you thinking? Here's some of the things I'm thinking. And the consultants that we have, granted, we can come along and help you do validation of a process. We can help you qualify equipment. We can help you deal with aseptic processing issues in traditional aseptic processing. But some of the things we do really well is we've got some really seasoned people, 25, 30, 35 years of experience that have been working in industry across many kinds of delivery forms, dosage forms, devices, drugs, biologicals. And those people have learned over the course of their career how to apply the regs and how to think about these things. And I look for those kinds of consultants to work on those kinds of projects. And those are the most interesting and rewarding and fun projects, frankly, because you're advancing medical science in a novel way that hasn't been done before. You're setting precedent. And as that company continues to work with FDA, you're helping to set the future of the regulations and the guidelines. It's a lot of fun.
I mean, I can hear how excited you are about it, and I think it's exactly what we're talking about with compliance architects, that you're not just bringing in another pair of eyes, you're bringing in another pair of eyes who has been working on these kinds of projects across a spectrum and have taken lessons from that they can apply and cross-pollinate. You're really helping different companies that are all trying to do the same thing learn from each other without necessarily having to talk to each other. They can talk through the learnings that you can share.
Correct. I think the other thing that's really interesting is the best consultants are people that really know how to use Google because the majority of the answers are out there. So in thinking about some of the things that I needed to do on one of the most recent projects I'm working on, I thought, surely somebody's done this before with a diagnostic assay, some of the things that I was trying to accomplish. And sure enough, I went and looked at the COVID literature for the PCR assay for COVID and looked at how they approach certain aspects of design control, and it was all there in academic research papers. So it's very interesting because when you want to try to bring together a set of traditional guidelines, standards, regulations, ISO, ICH, and then you have a very novel way you're trying to apply this, right? Sometimes if you go look at some of the academic research and some of the things people have done out there in hospital labs or academic institutions, you find some interesting approaches that people have applied that you realize when you read some of that and scan some of it, that's going to solve my problem. Here we go. So I think it's really important as you're doing this that you're not afraid to go into the literature and research, and you're not just going in and looking at guidelines and regulations and in a very rote way trying to apply them and apply rules. It's very important that you look at how people have approached, how do I standardize an assay? How do I standardize my cell culture medium because I'm trying to do this in this academic institution? While they may not have generated the paperwork that the FDA would approve, they've often done the work in a way that helped them substantiate their PhD thesis or do something else as a researcher. And some of the fundamentals have been figured out, and you just need to find them and apply them.
Fantastic. I know this has been a wide-ranging conversation, and I would love to just underline a couple of things that we want to make sure people are taking away from it. So if there were one or two bullet points that you want people to really think about further after listening to this episode, what would those be?
I think it's that, remember, guidelines and standards are written long after the technology's been introduced because the regulators have to learn the technology before they can write the guidance. So if you are in the leading edge, it's going to be your job to find the way. It's going to be your job to help educate the FDA and help them understand why what you've done answers the traditional questions that they ask. But don't look to the guidelines to answer all your questions for you because they will write a guideline about what you're doing right now, 5 or 10 years from now.
Fantastic. you mentioned it earlier, but I do want to give people an opportunity, if they've been listening to this and they can see the wealth of knowledge here, if they've got some questions, they have ideas they want to run by you, what is the best way to get in touch with compliance architects?
I would say to drop one of us an email. And so if you go to compliancearchitects.com, our website, you'll find the contact information for all of us. And if you even send a note to the general email box, it will very quickly be addressed. And Jack or Jerry or Jamie or I will get back to you right away.
Fantastic. And I always appreciate when people say contact me directly, because I like to think...
You can always contact me directly. Look me up on LinkedIn. I'm there.
I've been enjoying this conversation so much, but of course, I'm not a pharmaceutical executive. So when they come to you, they're coming to you with something really important. And it's such a generous thing to do. So first of all, Teresa, I want to thank you so much for your time today.
Thank you.
This has been so much fun. You've been joining us for another episode of Executive Platform's Blueprint podcast series. I've been Jeff Mix. Let's do it again soon.